Assalamu alaikum. Welcome to 5th International Conference on Applied Zoology 2022. I am Mehwish Murad Khan, MPhil scholar from Government College University, Faisalabad. I am going to moderate this session on the behalf of Applied Zoological Society of Pakistan. This session includes different interesting talks related to different topics such as natural product, microbiology, and disease management. Our first presenter is Dr. Razia Noreen from Government College University, Faisalabad, and her title of presentation is Study of Indigenously Produced Varieties of Moringa oleifera Leaves to Monitor its Antioxidants and Antimicrobial Activity. Please, Dr. Razia Noreen.
Assalamu alaikum, dear participants and organizers of 5th International Conference on Applied Zoology 2022. I am Dr. Razia Noreen, Associate Professor from Department of Biochemistry, Government College University, Faisalabad. Today, the topic of my presentation is the study of indigenously produced varieties of Moringa oleifera leaves to monitor its antioxidative and antimicrobial activity. Regarding the introduction, as the moringa is a very famous plant, and the leaves of moringa oleifera have highest ratio of iron, calcium, and vitamins. They have used for boosting the immune system. Very ancient time, the people also used to treat the ailments and different diseases. As it is reported in the literature, 100 gram of dry leaves of moringa oleifera contain highest amount of calcium, potassium, vitamins, and minerals than the food normally be taken. That's why it is preferably considered these days, since few years, a very good source of all these minerals and vitamins. And its popularity is also due to the presence of flavonoids and phenolics. Flavonoids helps in reducing the reactive oxygen species, whereas it also the presence of phenolics and flavonoids, they causes the antioxidant and antimicrobial activity. So we have used dry leaves of two varieties, Moringa oleifera, and these varieties were indigenously produced here in Faisalabad, and these were the Indian and Multan variety. And first of all, their leaves were collected, dried, washed, and clean, dry, shade dry, and then their powder was used for further experiments. And the extraction was done by using the sonication method. The ethanol and methanol was used as the solvent for this purpose. For the phytochemical analysis, first of all, the FTIR was performed for the both varieties, for Indian and Multan variety, and then HPLC was done to find the phenolics and flavonoids. In the end, the agar-well diffusion method was used to check the antibacterial activity, and then for antioxidant acid, DBPH, total phenolic contents and total flavonoid contents was studied. The FTIR results of Indica and Multan variety of Moringa oleifera shows the presence of amines, carboxylic acid, and aromatic acid with the slight variations. As the FTIR analysis, they are, we know this very well, they are very reliable to detect the various bonds and stretches which are present between the functional groups. And the data was obtained by using the frequency range of 400 to 500 to 4000 centimeter. And we have found that the most of the peaks in our results, they showed the major functional groups present in the Multan variety as compared to the Indian variety. And regarding the HPLC results in Multan variety, more compounds present that are responsible for antioxidative and antimicrobial property Whereas in Indian variety, the compounds responsible for antioxidative and antibacterial are present in less amount. Here we have the chromatogram which was obtained from the both varieties. This is the chromatogram of HPLC which was obtained by using the Multan variety of Moringa oleifera. And obviously the both varieties, this one is the Indian variety and this one is the Multan variety, both uh, showed the good results regarding the compositions of various phenolics such as gallic acid, catechin, chlorogenic acid, fumaric acid and the synaptic acid. They were found in the better quantity in the Multan variety as compared to this one which is the Indian variety. Whereas in Indian variety, we have observed fumaric acid and the caffeic acid in the good concentration. The peaks of HPLC chromatogram of both varieties uh, of the India and Multan, they showed the methanolic extract. By using the methanol extract, they showed the better results and showed that there are high contents of the phenolics and flavonoids are present. Regarding the antibacterial activity, the agar-well diffusion method, the four bacteria was used for experiments, and in these, the Staphylococcus aureus gave the best result activity as compared to the E. coli, Pseudomonas, and the Bacillus subtilis. And the DPPH assay, it revealed that the free radical scavenging activity, which was greater in Indian variety than the Multan variety of Moringa oleifera leaves, and total phenolic contents was found higher in the Multan variety than the Indian variety of Moringa oleifera leaves. Or total phenolic contents, they were higher in the Indian variety than Multan variety of Moringa oleifera leaves. So India and Multan variety, if we include these both varieties which are indigenously produced here in our botanical garden of university, these both varieties of the Moringa oleifera leaves, they contain the phenolics and flavonoids which were essential, which are normally essential for the body and used in the treatment of diseases which may be due to the bacteria and reduce the damage caused by the reactive oxygen species. So the presence of essential bioactive compounds make the moringa oleifera leaves as a potent antioxidant and antimicrobial agent. Our results showed the very good minimum inhibitory, I say, very good concentration of these amounts, which showed that they have the potential to use as the antioxidant agent also as compared to the commercially available antioxidants. Thank you. And if you have any question, then you are welcome.
Thank you, Dr. Razia Noreen, for such an informative talk. Our next presenter is Dr. Afshin Kamar from Qaeda-Azam University, Islamabad. And her title of presentation is Mechanical Strength of Novel Dental Restorative Material Incorporating the Titanium Oxide Nanoparticles Synthesized by Bacillus subtilis. Please, Dr. Afshin Kamar.
Bismillahirrahmanirrahim. In the name of Allah, the most beneficent and the most merciful. My name is Dr. Afshin Mansoor and I'm working as Associate Professor in School of Dentistry, Shaheed Zulfikar Ali Bhutto Medical University. I did my MPhil in Dental Materials from Rifa International University where I was awarded with the gold medal for my research. Currently, I'm a PhD scholar in Qaeda-Azam University. Today, I'm going to elucidate and shed some light on the topic of current and worldwide interest, titled Mechanical Strength of Novel Dental Restorative Material Incorporating the Titanium Oxide Nanoparticles Synthesized by the Bacillus subtilis. Let's have an overview of the current study. In the first step, the synthesis of titanium oxide nanoparticles utilizing the Bacillus subtilis will be carried out. In the second step, the characterization of these synthesized titanium oxide nanoparticles by using the standardized protocols will be done. In the third step, the mechanical strength of the titanium oxide nanoparticles incorporated in the dental restorative material will be done. Let's talk about the nanotechnology. Nanotechnology is derived from a Greek word, nanos, which meant dwarf because it works at the level of atoms and molecules. The size of the particles ranges between 1 to 100 nanometers in the nanotechnology. The nanotechnology is a highly emerging field currently because it is lighter, stronger, faster, more durable. That's why it is highly recommended in the medical and dental applications. Now, what is nanodentistry? The incorporation of nanoparticles in diagnosis, monitoring, control, prevention, and treatment is known as nanodentistry. Let's talk about the synthesis of nanoparticles. There are three basic routes of the preparation of nanoparticles worldwide. These are physical, chemical, and biological. The physical methods incorporate the utilization of tube, furnace, ceramic heater, ball milling, and laser ablation. On the other hand, the chemical methods utilize the sol gel, solvothermal, and precipitation methods. Last but not the least, biological methods incorporate the natural resources such as microorganisms and plants. Here the question arises, why is biological method considered as the best option for the synthesis of nanoparticles? The answer to this question is that the nanoparticles prepared by this route are easily available, eco-friendly, sustainable, non-toxic, cost-effective, and safe. That's why they are highly recommended for utilization in the medical field. Why is this study paramountly indispensable and cannot be ignored? There is a deficiency of cost-effective, biosafe, and biocompatible titanium oxide nanoparticles in the current era to be utilized in the dentistry. The aim of the current study is to develop a cost-effective, biocompatible, and biosafe novel dental restorative material with enhanced mechanical strength. This aim will be attained by carrying out the three main objectives: to synthesize the titanium oxide nanoparticles by Bacillus subtilis, to characterize the titanium oxide nanoparticles prepared by the Bacillus subtilis, and to incorporate the titanium oxide nanoparticles in the dental restorative material to produce a novel dental restorative material with enhanced mechanical strength to treat the dental caries. Let's talk about the basic methodology design. In the first step, the Bacillus subtilis will be incorporated to produce the titanium oxide nanoparticles. In the second step, these titanium oxide nanoparticles will be characterized by utilizing the X-ray diffraction spectroscope, diffuse reflectance spectroscope, atomic force microscope, scanning electron microscope, energy dispersive X-ray spectroscope, transmission electron microscope, and Fourier transform infrared spectroscope. At the end, these titanium oxide nanoparticles will be incorporated in the conventional dental restorative material to produce a novel dental restorative material, and then its mechanical strength will be tested by utilizing the universal testing machine. Let's talk about the basic methodology of the synthesis of titanium oxide nanoparticles. First of all, the Bacillus subtilis culture and nutrient broth of 100 ml will be taken. After that, the diluted culture solution will be kept and grown for 24 hours. After 24 hours, 80 ml of the diluted culture solution and 20 ml of titanyl hydroxide will be mixed and heated at 60 degrees centigrade for about 20 minutes. After 20 minutes, this solution will be kept in a shaking incubator at 28 degrees centigrade for about 24 hours. After that, this solution will be dried at 80 degrees centigrade for next 24 hours. After the drying, the titanium oxide nanoparticles will settle down at the bottom of the flask in the form of the powder, which will be calcined at about 450 degrees centigrade for 3 hours in a furnace. After that, the pure crystalline form of titanium oxide nanoparticles will be attained. Let's comment something about the results. The first result, which is visually observed, is the color change, which shows the preparation of titanium oxide nanoparticles. All of you can very well see that in the figure A, the solution is yellowish in color, which is the Bacillus subtilis culture solution with titanyl hydroxide. And in the figure B, you can see the preparation of the titanium oxide nanoparticles by the change in color to pure white. The X-ray diffraction analysis shows the mixture of the anatase and rutile phases, which shows that these titanium oxide nanoparticles are highly biocompatible because the mixture of the rutile and anatase phase are considered safe in the literature. The UV-based diffuse reflecting spectroscopy confirmed the formation of large particle size because the band gap energy value is less. As the literature says that if the band gap value is less than the standard value of 3.2, then the size of the particles is larger. The atomic force microscope results totally confirmed that these titanium oxide nanoparticles are smooth, having soft surface. That's why they can be easily incorporated and mixed in the dental restorative material. The scanning electron microscopy results confirmed the spherical shape of the nanoparticles with a large size, which you can also see in this sun picture. The energy dispersive X-ray spectroscope totally showed the pure peaks of titanium and oxygen without the involvement of any other impurity. Now, the transmission electron microscope results are totally in collaboration with the SEM and the TEM results because the size of the nanoparticles are quite large. The Fourier transform infrared spectroscope showed the presence of the functional groups which were safe from any sort of impurity and totally biosafe and biocompatible in the nature. All these results of characterization totally confirmed that the titanium oxide nanoparticles were biosafe and biocompatible to be utilized in the dentistry. Now, in this step, we will describe how the mechanical strength of this novel titanium oxide incorporated dental restorative material was enhanced. In this bar graph, you can easily see that the control group having no titanium oxide nanoparticles elaborates the low value of the strength as compared to the experimental group where the flexural strength is enhanced. In this next slide, the compressive strength analysis was done where again the control group without titanium oxide nanoparticles elaborates the lesser value as compared to the experimental group that was having the titanium oxide nanoparticles. It completely shows that the mechanical strength of the novel dental restorative material containing the titanium oxide nanoparticles was enhanced as compared to the conventional dental restorative material without titanium oxide nanoparticles. Now let's talk about the conclusion of the current study. The current study concluded that the novel dental restorative material containing the titanium oxide nanoparticles synthesized by the Bacillus subtilis were cost-effective, biosafe, and biocompatible with enhanced mechanical strength, thereby ensuring the longevity of the restoration in the oral cavity. And this novel dental restorative material can easily treat the dental caries quite effectively and efficaciously for the longer duration. These are the references of the current study. Thank you very much and Allah Hafiz.
Thank you, Dr. Afshin Kamal, for such a productive presentation. Next abstract is going to present by Dr. Sabah from Women University, Multan, and her title of presentation is Bacterial Biofilms, an Efficient Absorbent Material for Arsenic Remediation. Please, Dr. Sabah.
Bismillahirrahmanirrahim. Assalamu alaikum everyone. I am Dr. Sabah from Department of Microbiology and Molecular Genetics, the Women University Multan. And the title of topic that I am going to present today is bacterial biofilms and efficient absorbent material for arsenic remediation. This is the abstract of my paper that I am going to present today, but I will not completely describe this abstract over here because you already would have seen it in the abstract booklet of the conference. So let's start with introduction of the topic. Pollution of surface and groundwater bodies due to arsenic is one of the most important and life-threatening problems worldwide, with approximately 150 million people consuming arsenic-contaminated water. So this is a very large number of people, group of people that are consuming arsenic-contaminated water on the daily basis. And this arsenic is highly carcinogenic and poisonous toxic metalloid that is continuously going inside the human bodies, and we need to remediate this arsenic contaminated water. So let's see an approach of bioremediation. So bioremediation of arsenic through microbial biomass constitutes biosorption, that is an extremely efficient process due to low cost, reusable biomaterial and sorption of toxic metals and metalloids within short time periods. So this is the economic method and eco-friendly method by which we can remove the excess amount of arsenic from the contaminated water bodies. And one of the living methods is the use of EPS, that is extracellular polymeric substance, an essential component of bacterial biofilm. So let's see what is an EPS. It is a complex high molecular weight extracellular polymers that are secreted by microorganisms and mainly composed of hetero or homopolysaccharides, proteins, nucleic acids, lipids, and humic-like substances. So this is the composition of EPS, and different environmental factors such as carbohydrate sources, dissolved oxygen content, temperature, pH, metal ions, and shear forces, etc. may alter EPS composition, structure, as well as the properties. So EPS is affected by many different factors, and this may vary depending on the situations that are available to the bacteria in the surrounding environment. And EPS is generally present in two different forms that include soluble or colloidal EPS. It is excreted outside the cellular surfaces, and the second one is accumulated within microbial aggregates and tightly bound to the cells, known as bound or capsular EPS. So EPS produced during biofilm formation exerts several beneficial impacts on bacterial cells, including their protection from harsh environmental conditions like toxic metals and antibiotics, retention of water, and enzymatic activities. So this is basically biofilm formation is the protective action mechanism of bacterial cells to prevent them from harsh environmental conditions that may be in terms of some metals or in terms of some antibiotics or other harsh conditions. Moreover, changes induced by EPS in the bacterial cell surface, hydrophobicity, charge, and viscoelasticity help in cell adhesion and aggregation for biofilm formation. Bacterial EPS contains many different types of chemical compounds, predominantly acidic polysaccharides having ionizable functional groups like hydroxyl group, carboxyl, amide, sulfate, and phosphoryl groups that interact with toxic cationic metals and metalloids. These polymeric compounds present in EPS undergo electrostatic interactions with metal cations and therefore reduce their mobility or toxicity through accumulation or immobilization within EPS. So the EPS having different polymeric compounds actually immobilize toxic heavy metals inside themselves, and they serve as biosorbent material for removal of these toxic metals and metalloids from the environment. Immobilization of toxic metals and metalloids constitutes an important and promising strategy to alleviate the toxic metals from environment. Metabolism independent passive bioabsorption of toxic metals within EPS is more favorable than metabolism induced bioaccumulation inside the bacterial cells. Therefore, EPS has been investigated for the sequestration of toxic metals, including arsenic, due to its metal binding properties. So this study was aimed to determine the role of EPS as a suitable absorbent material for arsenic bioremediation. So we wanted to target arsenic, and this study was designed to actually use bacterial biofilm EPS as a biosorbent material that is economical and eco-friendly for the removal of arsenic from our environmental sources. Therefore, effect of arsenic toxicity on bacterial EPS production, chemical composition, and structural characteristics was studied through different colorimetric quantitative methods, Fourier transform infrared spectroscopy, and scanning electron microscopy. So now we will discuss the methodology that was followed during this study. First of all, there was acquisition or selection of bacterial strains. In this study, for the purpose of bioremediation, we used two strains of arsenic-resistant and biofilm-forming bacteria. These bacteria were Exigobacterium profundum PT2 and Ochrobacterium ciceri SW1. These two strains were previously isolated from industrial wastewater effluents, and they have been characterized as arsenic resistant as well as biofilm forming bacterial isolates. Secondly, these bacterial strains were checked for their slime production assay by using Congo red agar method. In this, the ability of bacterial strains to produce slime was detected qualitatively by Congo red agar assay. Congo red agar plates were supplemented with and without arsenate or arsenite, and these plates were prepared and bacterial strains were inoculated on these plates, and after overnight incubation, the colonies were checked for the production of black color that will indicate the production of slime or biofilm on the surface of Congo red agar medium. Thirdly, the bacterial biofilms were grown and their EPS was extracted by following the method in which bacterial pellets was treated with ethanol, and it was under the cold press method. Then morphological and biochemical characterization of EPS was performed, and in this method, EPS was extracted, and after extracting, its color, texture, and weight was recorded, and protein content was measured by Bradford assay, whereas total carbohydrate content was measured by using colorimetric phenol sulfuric acid method. Moreover, morphological characterization of EPS was also done through scanning electron microscopy, and scanning electron microscopy images helped us to identify different morphological changes that happened in EPS due to the stress of arsenate and arsenite. Moreover, surface characterization of EPS was also done through Fourier transform infrared spectroscopic method that helped us to identify different functional groups that were affected due to the stress present in the form of arsenate and arsenite. Now let's move towards the results. As we checked the slime production assay for both bacterial strains that were selected, it was shown that Exigobacterium profundum PT2 displayed slime production in the form of black colored colonies, both in the presence and absence of arsenic. So their slime production ability was not affected by arsenic stress. However, in the case of Ochrobacterium ciceri SW1, it did not produce black-colored colonies on Congo red agar plates, neither in the presence nor in the absence of arsenic stress. So, Ochrobacterium ciceri was unable to produce slime. Here you can see the red-colored colonies that are indicating negative results for slime production, whereas these black-colored colonies are showing positive results for the slime production. Now comes the morphological and biochemical characterization of EPS. If we talk about the morphological features and quantification of the EPS produced under arsenic stress, so let's see that in the case of Exigobacterium profundum PT2 bacteria, brownish powder was produced under controlled conditions. However, it turned into whitish powder under arsenate stress and grayish powder under arsenide stress. Whereas the quantity of EPS produced was maximum in the presence of arsenate stress, followed by in the arsenite, and minimum amount was produced in the control when there was no arsenic present in the growth medium. However, in the case of Ochrobacterium ciceri SW1, control strain also produced EPS that was less than that produced in the arsenate and arsenite stress. Now let's see carbohydrate and protein contents of EPS extracted from Exigobacterium profundum and Ochrobacterium caesari. So carbohydrate content was maximum in the presence of arsenate stress and then in the arsenite, and minimum was in the control condition. And similar trend was observed in the case of Ochrobacterium caesari. However, in the case of protein, the maximum amount of protein was observed in the presence of arsenite, followed by that in arsenate, and then control condition. And similarly, in Ochrobacterium caesari, maximum amount of protein was seen in arsenite, then in arsenate, and then in control condition. So we can say that protein concentration was increased under arsenite stress, whereas carbohydrate concentration in the EPS was increased under arsenate stress. This is the scanning electron micrograph of EPS produced by Exigobacterium profundum PT2 under arsenate and arsenite stress conditions. And we can see that in the control conditions, the biofilm EPS was having pores-like structures. These are pores when these are magnified to higher resolution. However, in the case of arsenate stress conditions, you can see that there are very large agglomerations and clumpings together. These are actually enhanced amount of biofilm and enhanced amount of EPS in the picture, that is showing the accumulation of different carbohydrates that we have also seen in the quantitative method, that carbohydrate content was increased under arsenate stress. So that is also evident from here in this picture. And now if we come towards arsenite stress, so the biofilm, the EPS is again very enhanced, but here EPS is having different structure, that is of proteins, like because we also run control samples of proteins and sugars along with these samples. So we compared them with the literature, and this is evident that protein content is high in the arsenite, whereas carbohydrate content is high in the arsenate stress conditions. These are the results of FTIR of Exigobacterium profundum PT2. And if we see this graph, then the peaks between the range of 800 to 900 per centimeter wave number, they are showing the presence of different glycosidic linkages. And in the control, the peak around 1211, whereas in the arsenate and arsenite treated, the peaks of 1218 and here 1134. These are also representing the polysaccharides and increased production in the arsenite as compared to control. So when we see the peaks in the range of 1700 per centimeter wave number, so these peaks are actually showing the presence of amide groups. And now we see the spectrum, FTIR spectrum of Ochrobacterium caesari SW1. So it peaks also around 847, 800, and 800, 847, and 880. These are showing the presence of glycosidic linkages. So we can clearly see that arsenate, the EPS of arsenate is having highest concentrations of all the functional groups present inside the bacterial EPS, whereas that of arsenite is less than arsenate, but still higher than control condition. So in this study we found that EPS produced by biofilms of both Exigobacterium profundum and Ochrobacterium caesari, they served as excellent biosorbent material for arsenic remediation, and we can further more characterize these bacterial strains and use them for some practical approach to remediate arsenic from the environment, and devise some biofilm based biofilter that will be used to treat water and remove arsenic from it. So this was all about the topic that I presented today. If you have any questions, you may ask. Thank you so much for your precious time and for listening me.
Thank you, Dr. Sabah, for nice talk. Next topic is presented by Dr. Samreen Gul Khan from Government College University, Faisalabad, and she is going to present Synthesis and Anti-Cancer Activity Novel 1,2,4-Triazole Derivatives. Please, Dr. Samreen Gul Khan.
Bismillahirrahmanirrahim. Assalamu alaikum everyone. I'm highly thankful to Applied Zoological Society of Pakistan and Khwaja Farid University of Engineering and Information Technology for providing me this opportunity to share my work with scientific community at 5th International Conference on Applied Zoology 2022. I'm Dr. Samreen Gul Khan from Department of Chemistry, GC University, Faisalabad. Today, the topic of my research work is synthesis and anti-cancer activity of novel 1,2,4-triazole derivatives. Triazoles are heterocyclic compounds containing 3 nitrogen atoms and 2 carbon atoms. Depending upon the position of these 2 nitrogen atoms, it has 2 different isomers, this 1,2,4 isomer and 1,2,3 isomer. 1,2,4-triazole is reported to have significant activities like anti-cancer, antifungal, anti-inflammatory, antimicrobial, antibacterial, etc. Here are some drugs available in market having 1,2,4-triazole common in them, like these are different antifungal drugs, and here is a list of different anti-cancer drugs and antiviral as well as anti-migraine. Here is overall scheme of my work. I took an organic acid and it was converted into its respective ester by reaction with ethanol and in the presence of sulfuric acid using as a catalyst. Then this ester was converted into its respective hydrazide, and this hydrazide was then cyclized into its respective triazole. Then this triazole was reacted with a series of different unsubstituted acetamides, and after successful reaction, they gave us a series of different triazole coupled acetamide derivatives. Here is a list of different 6 derivatives. They are different in position of these groups. I have attached some electron donating as well as electron withdrawing groups. Formation of compound was confirmed by their spectral data analysis. Like this, it's the proton NMR of the compound, is the aromatic region. You can clearly see different peaks for aromatic protons. Is the C13 NMR of compound. After successful synthesis, compound were checked for their anti-cancer activity against HepG2 cancer cell line cell. Results were very promising, especially compound 6B and 6D showed excellent activity with lowest IC50 value. This table showed percentage inhibition against HepG2 cell line cell of different 6A to 6F. We have used different concentration of these compounds. Is the graph showing percentage inhibition. You can clearly see that 6B and 6D were having promising results. Compounds were also checked through in silico modeling using c-KIT tyrosine kinase as an active protein. Here, compound 6D was found to have excellent result in silico modeling as well. So we conclude We have synthesized a series of 1 to 4 triazole coupled acetamide derivatives. All derivatives were checked for their anti-proliferative activity against Hep2 liver cancer cells. Among all derivatives, compound 6B and 6D containing electron-donating methyl substituent at different positions of aryl ring showed the best cell viability with lowest IC50 value of 13 and 12 micromolar respectively. Mechanism of inhibition of compound was further studied by in silico modeling, and all results were in accordance with our biological findings. So we conclude that compound 6B and 6D could be utilized as a lead compound in cancer therapy in near future. Thank you for your attention.
Thank you, Dr. Samreen Gul Khan, for an amazing abstract. Our next presenter in this session is Irish Atiq from Gujarat University, and her title of presentation is Characterization of Lactobacillus species from tiger and leopard feces and assessment of their pharmacogonistic properties. Please, Irish Atiq.
Assalamu alaikum. My name is Irishati. I am from Department of Zoology, Gazi University, Delhi. Topic is characterization of Lactobacillus species from tiger and leopard species and assessment of their pharmacognostic properties. Introduction. What are probiotics? Probiotics are living microbes, live microbes, in which different types of microbes occur, such as Lactobacillus, Lactococcus, Bifidobacterium, and Bulgarius, and so on. As I told you that my topic is on Lactobacillus. Lactobacillus are lactic acid bacteria. They produce lactic acid. They are found in our diet. Many types of lactobacillus are found in animal intestine, animal body, and it has so much beneficial effect of animals. Objective of this study was to isolate and characterize Lactobacillus species from fecal matter of tiger and leopard to check and compare probiotic tolerance such as pH tolerance, NaCl tolerance, and bile salt tolerance. To investigate in vitro pharmacological applications such as antioxidant property, anti-inflammatory activity, and antimicrobial activity. Material and method. We collected our samples from fecal matter of tiger and leopard of Lahore Zoo, Punjab, Pakistan. After collection, we put it in Pepton water for 24 hours. After that, we put it on media. Media was used MRS with cysteine. Cysteine is good for the growth of lactobacillus, and calcium carbonate is also good to identify the lactobacillus species. As I told you that lactobacillus are lactic acid bacteria. Lactic acid bacteria use calcium carbonate and utilize calcium carbonate, and we can identify by that these are lactobacillus. Bacterial isolation, purification, and identification are done by morphology, biochemical, and some potential tests. Morphological characterization, colony morphology and cell morphology. For identification of lactobacillus, we first of all we have to identify its colony. It has when we collect different microbes, when we see different microbes and we want to collect lactobacillus, then there are so many colonies. We have to identify the colony of lactobacillus. We have to identify cell morphology by different staining mechanisms such as gram staining, spore staining, and acid-fast staining, gram staining. Gram staining method is done by Colin et al. 2004. In this, we use the method followed by Colin et al. 2004. in which crystal violet, iodine, and alcohol is used. Safranin is used as countercurrent stain. Spore staining. Spore staining method is followed by Harley and Prescott 2002. In this, first of all, malachite green is used as primary stain. After that, heat it and decolorize by using water. In the end, we use counter stain safranin to see that spores formation occur or not. In lactobacillus, there is no spore formation occur. So this test will help us in identification of lactobacillus species. Acid fast stain. This method is followed by Facing and Verma 1998. In this, cardboard system is used. After that, heating processes is done. Then acid alcohol is used to decolorize, and as countercurrent stain, we use methylene blue. Biochemical characterization is also used to identify and characterize lactobacillus species. Catalyst test. This test is done by hydrogen peroxide, 3% hydrogen peroxide. If a bubbles formation occur, then we can say that these are not lactobacillus species because lactobacillus have no ability to produce gas. Carbohydrate fermentation test. This test was done by glucose and mannitol. Potential test, NaCl tolerance followed by Hawk et al. 2010. pH tolerance test done by Hawk et al. 2010. Biocide tolerance is done by using the protocol described by Celia and Maria 2001. Here is some activities, some pharmacognostic activities, antimicrobial activity, antioxidant activity, anti-inflammatory activity. Antimicrobial activity is done by a well diffusion method in agar plate. It is done against three microbes such as E. coli, streptococcus. Antioxidant assay or antioxidant activity, we done some three assays and three activity to see that the lactobacillus are antioxidant or not, or our strains are antioxidant or not. First one is DPPH, free radical scavenging activity. This method is done followed by Reshma et al. 2014. Hydroxyl radical scavenging activity. This method is done by De Eveler et al. 2004 and superoxide anion radical scavenging activity. This method is estimated by Pyrogalore auto-oxidation method, Chow et al. 2003. At the end, we done anti-inflammatory assay. In this, inhibition of albumin denaturation occur. This method is followed by Reshma et al. 2014 with some modifications. Modifications was done by supernatant and pellets. We did this experiment on pellets and we made in this cell suspension. Here are some results. Colony morphology. Lactobacillus, we isolated two strains. One is Lactobacillus from tiger and second is Lactobacillus from leopard fecal matter. We collected two strains and both strains have same colony morphology. Colony morphology was same. After sequencing, we know that both species are same, but the sources was different. Here is the result of biochemical characterization. It's gram staining result. In gram staining, the lactobacillus gives purple color because of the crystal violet, because it is gram-positive bacteria, so it gives it gives purple color. Non-spore forming stain. Here we can see that both lactobacillus, the tiger and leopard, both have no spore formation. Both have no green color spore. Both have vegetative cells which are red in color. Acid fast staining. In acid fast staining, you can see that they give blue color, which means they have no cells like that which can produce tumor, which are non-acid fast. Lactobacillus are acid fast. Lactobacillus are acid fast strains. So by these three staining, we can say that we can identify that our strain may be lactobacillus. Negative catalase test. If catalase test is negative, it means if bubble formation is not occurring with H2O hydrogen peroxide, it means these are lactobacillus. Lactobacillus because produce any type of gas with hydrogen peroxide. Next is mannitol utilization and glucose test. In mannitol utilization, you can see that control is in red color. When we put strain in it, when we inoculate it with strains, then it converted mannitol into glucose. It utilized the mannitol and convert it into yellow color. Gas production and glucose test. In this, we use glucose instead of mannitol, and we use Durham tubes. Durham tubes are for identification of gas. You can see that here some bubble formation is occurring, some gas you can see in L and T tubes. There are some gas. It means lactobacillus. This type of lactobacillus, which is collected, which we isolated from fecal matter of leopard and tiger, are gas producing lactobacillus. Here is potential test result. Potential test, we done 3 types of potential tests: pH concentration, NaCl concentration, bile salt concentration. Lactobacillus are mostly are of different types, and they can they can survive at different pH concentration. You can see that both are giving same concentration of on 2, but in this blue color is for strain T, that strain which is isolated from tiger, and this orange color is for that strain which is isolated from lion. So here is the result. You can see that the strain T and the strain L both are giving good production at pH concentration 6. Next is NaCl. In NaCl, the good production is on 2. NaCl, we use 2% of NaCl. So lactobacillus give good production, good growth on 2%. Same in bile salt concentration. The bile salt concentration is different because of the different sources. These 2 lactobacillus, in which strain T lactobacillus, the lactobacillus which are isolated from tiger, give good concentration, good growth on 0.2% bile salt concentration, while red one, the strain of leopard, is giving good growth on 0.4% bile salt concentration. Here is the antimicrobial assay. Both lactobacillus give antimicrobial activity against Streptococcus, E. coli, and Staphylococcus. It is the DPPH radical scavenging activity. Leopard strain gives 33% and tiger strain gives 18% activity against DPPH. Same as in OH ion scavenging activity, the leopard is giving 74% OH ion scavenging activity, while tiger strain is giving 50.6%. And in superoxide radical scavenging activity, both are giving approximately 36.6 and 38.6% activity against superoxide radical scavenging activity. There is not much difference in the activity of both strains. Next is anti-inflammatory assay, in which we done this method to check the inhibition property, to check that protein can be inhibited by denaturation. So in this you can see that the strain which is isolated from leopard is giving high anti-inflammatory activity such as 54%, and tiger strain is giving 17.3%. Conclusion: the strains which are isolated from fecal matter are characterized as lactobacillus species. These are indicated as probiotic tolerant. and these are identified as Lactobacillus brevis by gene sequencing after this research, after this study, and both Lactobacillus brevis showed pharmacognostic properties but with different percentage. This study enhances the status from probiotic to superbacteria with antibacterial, antioxidant, and anti-inflammatory capabilities. In three ways, this will resist us to cope up and preventing a variety of disease. Thank you so much.
Thank you, Irish Atiq, for astounding presentation. Next abstract is presented by Asif Nazir Memon from University of Sindh, Jamshoro, and his title of presentation is Biodiversity of Short-Horned Grasshoppers from Middle Sindh, Pakistan. Please, Asif Nazir Memon.
Auzubillahiminash-shaytanir-rajeem. Bismillahir-rahmanir-rahim. Welcome to International Conference on Applied Zoology, which will held in the Khwaja Farid University of Engineering and Technology Sciences, Rahim Yar Khan, in the cooperation with the Applied Zoological Society of Pakistan. I am Asif Nazir Memon, PhD scholar, Department of Zoology, University of Sindh, Jamshoro. I will deliver my oral presentation on the biodiversity and taxonomy of longhorn grasshoppers and shorthorn grasshoppers from the district Dadur Sindh. These are the contents of my study: introduction, objectives, material methods, results, conclusion, references, publication, and acknowledgement. First of all, introduction to the biodiversity. The biodiversity may be defined as a biological variation and variability among the organisms. The biodiversity term was given by Walter G. Rosen in 1985. More formally, biodiversity is comprised of several levels, starting with genes, then individual species, then communities of the creature, and finally, an entire ecosystem. Introduction to the taxonomy. Taxonomy is the knowledge about the classification, identification, and distribution of the various organisms to various groups. This term is derived from the Greek word taxis, means arrangement, and noms means law. The taxonomy is therefore methodology and principles of systematic biology and set up arrangements of the different kinds of organisms on the earth. Grasshoppers are the potential pest of all plants, trees, and most serious enemies of the agriculture. Moreover, grasshoppers attack not only the agriculture crops but also rangeland fields, and they cause considerable loss to our valuable crops. The short-horned grasshoppers are common in moist and swammy areas. These serious pests have been recorded from every part of country throughout the year. Some grasshoppers cause significant damage to the tree seedlings and agriculture crops. They are widely distributed and absent only in truly polar areas and some few small group of islands of the oceanic origin. They are hemimetabolous insects, means having incomplete metamorphosis. They are included in oligophagous and mixed feeder. They are functionally very important ground invertebrates in grassland ecosystem. Many species are green or straw colored, which help them to blend in their surroundings. They are herbivorous and include some of the most destructive agriculture pests known. A female short-horned grasshopper lays about 100 eggs in the soil. The eggs hatch after a rest period, and newly hatched names, miniature replicas of adult, pass through a series of mold before they become adult. And the series of change is called metamorphosis, and I already told you about the metamorphosis occurs in these insects, which is incomplete metamorphosis. They are often the main invertebrate in grassland ecosystem for consumers, and they are also important food source for the many group of predators, for example birds and others. This is the general structure of short-arm grasshoppers, including head, thorax, abdomen, mesothorax, the antennae, the protosternum, mesosternum, pronotum, and all other these parts of the body. These are the objectives of my study: to collect the large number of short and long arm grasshoppers from the different selected localities of the district Dadu, to identify the collected material up to subfamilies, genera and species level, and new record and new species. To prepare the taxonomic key for the subfamilies, genera and species. To prepare photographs with the illustrations. The material methods. This is the map of district Dadu, and district Dadu includes the 4 Talka Meher, Talka Kensha, Talka Johi and Talka Dadu. So this map showing the 4 Talkas of the district Dadu. This is the methodology. The specimens were brought to Advanced Entomological Laboratory Department of Zoology, University of St. Jamshoro, and Zoological Lab of Government Ustad Bukhari Degree College, Dadu. The specimens were examined in the lab, samples pinned, and examined their important morphological characteristics such as head, including antennae, thorax, pronotum, prosternum, mesosternum, and abdomen, and other the main important parts of the body. This is the methodology. First of all, collect the specimens, then kill by the killing process, and then pinning, then stretch the specimens on the stretching board, then preservation takes place, and after the preservation the examination takes place. This is the list of equipments which I used during the experiments during my study. These are the list of chemicals which are used in the sample preservation: chloroform, naphthalene balls, alcohol, and killing insect jar. This figure showing the study area during the sample collection. The results of my study. There are 247 specimens collected from different areas of district Dadu. The specimens were identified into family activity, the 7 subfamilies, 12 genera, and 15 species. So this table showing the classification of these specimens into family, subfamily, genus, species. The family activity, these are the subfamilies: Oxini, Hemiacrididae, Podipodini, Melanopodini, Acrididae, Gamosphonae. These are the families, these are the genus, and these are the species which I collected during this table showing the collection of species from various localities of the district Dadu. This table showing the highest number of these species. Oxia hyla reported. The Oxia hyla reported is the maximum number in the selected localities: village Fakka, village Qazi Arab, village Muhammad Ibrahim Panwar, village Ghulam Hussain Gadihi, and village Bachalpur. The least number of the species which I collected from during the study, which is Enacrydium recipientum. This graph showing the collection of species from various localities of district Dadu. This table showing the incidence of the short-arm grasshoppers from various localities of district Dadu. The present study is carried in 2021 and 22. Percentage-wise data is presented as below. The highest percentage was recorded for the Oxia hyla, which is 19.02, and the lowest percentage was recorded for the Enacrydium recipientum, which is 2.02%. This is the graph showing the percentage of species found. As a conclusion, as a result of present study during the 2021, I have collected 274 species, sorry, 247 species specimens. These specimens belonging to family Acrididae, short-horn grasshoppers. During the present study, 15 species are described. The most abundant species is Oxia hyla hyla cervidi. Of the total number of all species, followed by Oxia phosphata, Herocleptus purpurata, Ilopus thalassenius, Ilopus thalassenius stimulus, Locusta migrata, Spenicus cervici, Trilophidia annulata, Acrita expilata, Honesta runditata, Inacridium rispinium, Elithera olipex, Eclotylus insabricus, Acrotylus fisteri, and Melanopilus differentius. Oxia hyla hyla is considered as a major pest of rice and found in all selected localities of the district Dhaka. So these are the key of the subfamilies of Acrididae. More keys. These are the publications. This is the research paper published in the Journal of Animal Sciences, University of St. Jamshoro. This paper published in the Journal of Global Biosciences. Another publication. These are the certificates. More certificates. The certificate from the Zoological Congress. These are the abstracts. References. More references. Acknowledgement. I am thankful to Honorable Dean Faculty of Natural Sciences, Professor Dr. Wazir Baloch, and the respected chairperson and my supervisor, Professor Dr. Naid Baloch, and my respected co-supervisor, Dr. Rifat Sultana, and all the respected teachers and research fellows of the Department of Zoology University of St. Jamshoro for their kind cooperation and guidance during my study. So I am thankful to all. Thanks a lot.
Thank you, Asif Nazir Memon, for such a descriptive talk. Kainat Razik from Cholistan University of Veterinary and Animal Sciences is going to present her abstract, and title of her abstract is Prevalence of Hepatitis B and C in District Bahawalpur. Please, Kainat Razik.
I am Kainat Razik, student of MPhil Zoology from Cholistan University of Veterinary and Animal Sciences, Bahawalpur. Today I am going to present my abstract entitled Prevalence of Hepatitis B and C in District Bahawalpur. Hepatitis is basically a Greek word. Hepa means liver and titus means infection. So hepatitis is an inflammation of liver tissue. Hepatitis can be caused by viruses, bacteria, drugs, and medicines. Viral hepatitis is of 5 types: hepatitis A, hepatitis B, hepatitis C, hepatitis D, and E. Both hepatitis A and E are self-restrictive and can be cleared by body's immune system, and can be caused by taking unhygienic food and drinking contaminated water. While hepatitis B is called serum hepatitis, hepatitis C is bloodborne in nature, and hepatitis D affect only those people who are already infected with hepatitis B virus. So hepatitis D is also known as superinfection. Hepatitis B virus is a DNA virus with a genome size of 3.2 kb, while HCV is an RNA virus with a genome size of 9.6 kb. Modes of transmission of the disease: both hepatitis B and C spread by direct blood transfusion, seminal and vaginal fluid, unsterilized use of unsterilized equipment, of medical equipment, dental equipment, and barber equipment, reuse of needles, both intravenous and intramuscular injection, physical contact with the infected person, hemodialysis, etc. There are some facts about prevalence of hepatitis B and C. Recently, WHO set a goal to eliminate the viral hepatitis by 2030. Viral hepatitis has a great death and disease burden due to liver cirrhosis and hepatocellular carcinoma. Both hepatitis B and C resulted in 1.38 million deaths annually. If we talk about the hepatitis B virus affect 257 million people, which means 3.5% of the total population of the world infected with hepatitis B virus. While 71 million people are infected with HCV, which means 1% of the total prevalent population of the world infected with hepatitis C virus. If we talk about the situation in Pakistan, 12 million people are infected with hepatitis B and C virus, which means 5% of the population of the Pakistan is suffering from hepatitis B and C, and annually 150,000 new cases are reported. There are some countries who are highly endemic for HBV and HCV. Pakistan and Egypt are highly endemic for HCV, while Somalia and Sudan are highly endemic for HBV. If we see the total prevalence and total disease burden, Pakistan and Egypt contribute to 80% of the disease burden. So keeping in mind in these facts, study was planned to determine the prevalence of hepatitis B and C in Bahawalpur. To know the current disease status and various risk factors were also recognized. Blood samples were collected from the patients visiting Bahawalpur Victoria Hospital. Duration of study was 6 months from December 2021 to May 2022. Around 263 samples were collected, including 81 males and 182 females. Meanwhile, questionnaire was filled by direct interview of the patient. Patient was asked about 13 questions to determine the prevalence, estimate the reasons of hepatitis B and C in Bahawalpur. These questions were about their age, gender, previous disease history, marital status, occupation, economic status, education, hospitalization, surgery, dental treatment, barber services, needle injury, blood transfusion, and drug abuse. After collection of a sample, samples were then transferred to a lab, and they were centrifuged at 3,500 rounds for 15 minutes. ICT and ELISA were the tests that were utilized to determine the positive ratio within blood samples. For this purpose, ICT kits that were utilized were, and for ELISA, autobiodiagnostic ink were utilized. After getting the results, statistical analysis was done using chi-square test with the help of IBM SPSS software version 22. Results shows that out of 263, 83 individual, which means 30.7% people were tested positive for hepatitis B and C, and out of 83, 67 were tested positive for hepatitis C virus, 14 were tested positive for 50, which is 5.3% for hepatitis B virus, and 2 were co-infected, which is 0.76%. However, the study shows a significant association of the disease with age, which shows that middle age group were more infected with hepatitis B and C, which is 31 to 40 years old people. Significant association of the disease were related to education, hospitalization, and dental treatment. In conclusion, we conclude that people who visited hospital frequently, received any medical care, including dental treatment, were illiterate, were more susceptible to infection. This study were also found that illiterate people are more infected due to the practice of unhygienic condition. The study will be helpful to implement the strict legislative measures to control the increasing infection of hepatitis B and C within Bahawalpur city and in the country as well. The result of the study will be helpful to formulate policies and adopting a preventive measures to control the increasing incidence of hepatitis B and C. At the end, there are some references, and thank you for your attention.
Thank you, Kainat Razik, for nice talk. Our next presenter is Gule Adan from University of Agriculture, Faisalabad, and her title of presentation is Effects of ciprofloxacin and sulfamethoxine on the activity of antioxidant enzymes of Mori Sirhinus mirigala. Please, Gule Adan.
Assalamualaikum, my name is Guledan. I have done my MPhil from University of Agriculture, Faisalabad. My topic of presentation is about effect of ciprofloxacin and sulfamethoxine on the activity of antioxidant enzymes of Mori. Basically, my topic of presentation is about effect of antibiotics on the activity of antioxidant enzymes of fish. When we talk about antibiotics, we basically say that antibiotics are used in human and veterinary medicines to treat or prevent the bacterial diseases. But the unnecessary and overuse of antibiotic is always harmful because they are not entirely removed from the wastewater treatment plants. The wastewater from the hospitals and direct manufacturing plants contribute to the presence of antibiotics into the water bodies. They adversely affect the non-targeted species such as the aquatic life. They are also considered as an important group of environmental contaminants because they can induce the oxidative stress which affect the antioxidant and detoxification processes of the fish. These changes can compromise the fish immunity which ultimately leads to the cellular damage. Consumption of fish treated with antibiotic also causes the human health risk. There is a diverse range of antibiotic and my research is on ciprofloxacin and sulfamethoxine. Both belongs to the different groups of antibiotic. Ciprofloxacin belongs to the fluoroquinolones group, whereas sulfamethoxine belongs to the sulfonamide group. Both are commonly used. Ciprofloxacin is used against the earache, bronchitis, and sinuses problem, whereas sulfamethoxine is used against gastrointestinal and respiratory tract infections. Both has tendency to generate the oxidative stress in the fish when used in excess amounts. When we talk about antioxidants, antioxidants are the molecules that are capable of stabilizing the free radicals. Free radicals are basically the highly reactive unstable molecules that has a lone pair of an electron, and in normal body, antioxidant enzymes have capability to deactivate and stabilize the free radicals by reducing their energies. When the normal balance between the antioxidant enzymes and the free radicals is lost, there is an oxidative stress, and the oxidative stress ultimately leads to the cellular damage. As we already know that aquaculture is a major economical sector and providing a lot of job opportunities along with meeting the growing demands of food, so that aquatic pollution is a serious and growing nuisance for the aquatic life because it increases the mortality of fish. Also, I want to develop the better insight about the toxicological effect of antibiotics on the fish. The main objectives of my study was to determine the effect of ciprofloxacin and sulfamethoxine on the GST and catalase enzymes of Mori. First of all, I collected the fish samples from the Fisheries Research Farm at University of Agriculture, Faisalabad. After the collection of fish samples, I acclimatized the fish samples in glass tank for about 10 days so that they adapt into a new environment. After the acclimatization, the fish was exposed to the different concentrations of antibiotic. The fish were divided into the three groups for both antibiotics, which were T0, T1 and T2. T0 was basically the control group where no antibiotic was added. T1 was the treatment group exposed to the low concentrations of antibiotic, whereas the T2 was the treatment group which was exposed to the high concentrations of antibiotic. After the acute exposure of 96 hours, fish sampling was done. Fish sampling was done after each 24 hours and fish was dissected with the help of dissecting box and the targeted organs such as the liver and gills were extracted out. After the collection of targeted organs, the homogenization was done and the organs were subjected to the antioxidant enzyme analysis. For catalase, first of all, we prepared the buffer solutions and then note the absorbance of catalase enzyme at 240 nanometer with the help of the spectrophotometer. For catalase enzyme, we prepared the buffer substrate along with glutathione solution and note the absorbance of GST enzyme at 340 nanometers. After recording the readings, the statistical analysis were done and results were generated. According to my results, catalase and GST activities increased significantly in the liver when exposed to the different concentrations of antibiotic, while in the case of gills, CAT and GST activities decreased significantly when exposed to the different concentrations of antibiotic. Variations in the antioxidant enzymes were observed, which were in following order. The liver were more affected as compared to the gills because liver is the main organ for the detoxification. Maximum activity was observed at 96 hours, and GST enzymes were more sensitive to antibiotics as compared to catalase enzymes. Exposure to a different concentration of antibiotic caused variations in the antioxidant enzymes of fish. We say that when fish was exposed to the different concentrations of antibiotic, it affects the antioxidant enzyme activity. Variations in the antioxidant enzyme activity could be considered as a reliable biomarker for the antibiotic studies. And that's it. Thank you so much.
Thank you, Gule Adil. Our next presenter is Dr. Muhammad Farhan Nasir from University of Education, Lahore, and his title of presentation is Effect of Organic and Inorganic Arsenic as2O3 on Hematological Parameters of Freshwater Air-Breathing Fish Sirhinus mirigala from South Punjab, Pakistan. Please, Dr. Muhammad Farhan Nasir.
Bismillahirrahmanirrahim. In the name of Allah, most gracious, merciful. I am Dr. Muhammad Farhan Nasir from University of Education, Lahore, Pakistan. The topic of my research is effect of organic and inorganic arsenic oxide on hematological parameters of freshwater air-breathing fish Sirhinus mirigala from South Punjab, Pakistan. The present study aimed to assess the survival response in terms of hematological changes of Sirhinus mirigala exposed to arsenic and arsenic trioxide in laboratory condition. In this investigation, the oxidative biomarkers in the blood sample of freshwater fish Sirhinus mirigala exposed at sublethal level of arsenic. Arsenic is included in the naturally occurring element and it is existed all over the world and more in the natural environment. Inorganic arsenic is easily absorbed in man by the oral route. In humans receiving orally less than 10 mg prevalent inorganic arsenic, the absorption rate was found to exceed 80%. Arsenic occurs in the environment in inorganic and organic form. Arsenic is existed in the trivalent form, which is more toxicity than the pentavalent form of arsenic. The trivalent form of arsenic is more soluble in aquatic environment. Many such compounds like arsenic produce in the environment. Environmental toxicity and these environmental toxins can induce physiological and biochemical changes in fish and lead to inhibition of growth and death in case of acute poisoning. Many such compounds like arsenic produce the environmental toxicity and bioaccumulation of arsenic in the blood and the body of fish may cause the infertility in the fishes. It may also cause the liver damage and cell death of fish. The freshwater of fish, Sirhinus mirigala, was collected from the ponds located in Muzaffargarh and Dera Ghazi Khan region of South Punjab, Pakistan. The size range of fish samples was started from 10 gram to 500 gram with length of 15 centimeter to 25 centimeter. The fishes were acclimatized with 1% KMnO4 to remove all pathogens from the surface of fish body. The fishes were maintained in the aquaria at a room temperature 28 degrees centigrade and the fish were introduced into 10-liter capacity containers of water containing specific metal arsenic. After the recovery period, all such treated fish were separated from the experimental container and blood samples were collected from 3 experimental individuals from each group and each time with an interval of 24 hours during the experiment. Blood of the fish was collected by puncturing the blood of fish and collected in the EDTA vial, and the samples were analyzed by the hemocytometer to get the values of red blood cells, white blood cells, hemoglobin, and other blood parameters. Results: chemical situation of the experimental tank. The temperature was maintained between 22 to 29 degrees centigrade. The light period, there was 11 hours of day and 13 hours of night. Alkalinity was 150.6 to 176.8 mg per liter. And the water hardness was maintained between 176 to 202. Chemical properties of arsenic and hydroxide. The melting point of arsenic is 36 and its density is 5.7. While arsenic trioxide, its boiling temperature is 869 Fahrenheit, while melting is 594 Fahrenheit. While arsenic pentoxide, its boiling temperature is 315 degrees centigrade and its density is 4.32 gram per centimeter cube. Different hematological components under variable sublethal concentration of arsenic in Sirhinus mirigalia. The total RBC in control group in the first 10 days it was 3.56, at 20th day it was 3.48, and at 30th day it was 3.52. While with 10% SLC it was 3.11, 2.72, and 2.14. While with 30% SLC it was 2.51, 1.95, and 1.52. If we talk about the white blood cells in control group, at 10th day it was 10.56, at 20th day it was 10.37, at 30th day 10.52. While with 10% it was 12.61, 14.52, and 16.37. While with 30% it was 13.66, 14.95, and at 30th day 17.82. And if we talk about the hemoglobin in control group, it was 8.51, 8.61, and 8.56. While with 10%, it was 6.79 at 10th day, 6.14 at 20th day, and 5.31 at 30th day. While with 30% SLC, it was 4.18 at 10th day, 3.21 at 20th day, and 2.64 at 30th day. The WBC count of Sirhinus mirigalia was increased with the increasing concentration of arsenic, and the increase in the number of WBC was found to be 29.36 to 44.17 and 69.39% at 10, 20, and 30th day exposure at 30% sublethal concentrations, respectively. The hemoglobin content of Sirhinus mirigalia in 10 and 30% sublethal concentration showed decreasing trend with a significant reduction of 20.21, 28.69, 37.97, and 50.88, and 62.71, and 69.16 at 10th, 20, and 30th day of exposure, respectively. Here in the figure, you can see the exposure period with 10th day, 20th day, and the 30th day. The mortality percentage with the increased concentration of arsenic Sirhinus mirigalia, as you can also observe that as the concentration of the arsenic was increased, the mortality rate was also increased. So it means it have a very lethal effect over the growth of the fish as well. Continuous exposure of lethal elements cause the increase of red blood cells in the fish and then sudden decrease in RBCs. With the decrease in RBC, the HP level of the blood was also decreased. Multiple forms of hemoglobin allows fish to adjust more efficiently to physiological stress such as varying water temperature and oxygen concentration. The increased concentration of white blood cells in this study was because of having direct activation of the defense system of the fish by having under the influence of any toxic element. Leukocyte is directly proportional to severity of stress condition in maturing fish and is a result of direct stimulation of immunological defense due to presence of heavy metals in wastewater pond. This is correlation with the report of ceravinin in the freshwater fish. The increased concentration of white blood cells in this study was because of having direct activation of the defense system of the fish by having under the influence of any toxic element. Leukocytosis is directly proportional to severity of stress condition in maturing fish and is a result of direct stimulation of immunological defense due to the presence of heavy metals in wastewater pond. This is in correlation with the report by ceravinin in freshwater fish. The toxicity of arsenic on the hematological parameters such as hemoglobin, RBCs, and WBC were significantly decreased because the arsenic affect the animal and led to anemia. It has also reported that pollutants generally cause change in the hematological parameters of the fish. Iqbal et al. Have also observed that reduced level of RBC, HB, and PCV in the Sirhinus mirigalia on the fish treated with lead. The MCH and MCHC increase in the blood of Sirhinus mirigalia exposed to nickel. The observed amount of MCH and MCHC indicate the hydrochronic microcytic anemia. The reduction in RBC count and HB content after 30 days of exposure indicates anemia associated with erythropenia. Our results are similar to the earlier reports of these scientists. Cinnarinus minigella exposed to arsenic concentration showed fall in number of lymphocytes due to decrease in non-specific immunity of fish. Leukocytes are involved in the regulation of immunological function in many organisms, and increase in the white blood cells in stress animals indicate a protective response to stress. The decrease in white blood cells count during acute and sublethal treatment may be due to toxic effects in head kidney of fish being primary site of hematopoiesis. Blood parameters were not affected when adult lake whitefish exposed to 0, 1, 10, and 100 microgram of arsenic gram per food. Low levels of arsenic toxicity fishes cause reduction in red and white blood cells. It is also reported a decreased level of hemoglobin and packed cell volume, that is PCV, in Clarice petricus exposed to arsenic. Cinnarinus minigella, when exposed to arsenic, showed decrease in granulocytes, erythrocytes, hemoglobin, hematocrit values. Contamination of water bodies by arsenic and its compound is a big threat to all the aquatic organisms, especially fishes. They enter inside the body where skin, gills, gastrointestinal tract, and accumulated in vital organs, and once carried by the blood. Aquatic organisms accumulate arsenic mainly as inorganic form, but few organisms like bacteria, phytoplanktons, etc. transform inorganic arsenic species into organic forms. Once toxic elements like arsenic may enter inside the body via food and drinking water, they produce deleterious effects on organisms. Government of different countries should monitor anthropogenic sources which are contaminating aquatic body and increase public awareness. Thank you so much.
Thank you, Dr. Muhammad Farhan Nasir, for your nice talk. Next abstract is presented by Dr. Muhammad Mudassir Shahzad from University of Education, Lahore. And his title of presentation is Effect of L-Malic Acid Supplemented Oilseed Meal Based Diet on Overall Performance of Common Carp Fingerlings. Please, Dr. Muhammad Mudassir Shahzad.
Bismillahirrahmanirrahim. All dear audience, I'm Dr. Muhammad Mudassir Shahzad from University of Education, Lahore. First of all, I am very thankful to ICAZ and Government College University, Faisalabad for hosting this mega event in Khaja Farid University, Rahim Yar Khan. The topic on which today we will discuss is about the effects of L-malic acid supplemented oilseed meal based diets on overall performance of common carp fingerlings. The topics on which today we will discuss are the abstract, introduction, methodology, and results. If we talk about the abstract, as you know, that the global exponential growth in human population has resulted in decline in the food availability. Thus, production of protein-rich aquatic food is high on agenda. This study was aimed to evaluate the potent effects of L-malic acid aggregation in coconut meal on body composition, nutrient utilization, blood hematological indices, mineral absorption, and ultimately on growth of Cyprinus carpio fingerlings. There were the 6 test diets that were designed in this experiment, in which L-malic acid was supplemented with a ratio of 0, 1, 2, 3, 4, and 5 gram per kg. And the feed pellets were prepared by using 1% of chromic oxide as an indigestible marker. Acidified diets basically helps in lowering the pH of fish gut. After the analysis of these results, we were able to investigate that the fish fed on 3 gram per kg organic acid will be very beneficial for better growth and better nutrient digestibility, better mineral absorption, and other parameters. If we talk about the introduction, first of all, we will discuss about the aquaculture. Aquaculture is basically a rapid flourishing sector of protein source that represents about 47% of global fish production. If we talk about the recent trends of intensive culture, it basically requires processed feed, but feed accounts about 50 to 60% of total expenditure. On the other hand, the basic major protein source in fish feed was the fish meal, but fish meal is statistics due to statistic supply of fish meal, there is a high cost of fish meal that is basically beared by farmers. On the other hand, we are searching for alternative plant protein sources that will be lower, that will be available throughout the year at lower cost, but having good protein contents. From these protein sources, we have analyzed that coconut meal, sesame meal, sunflower meal, and soybean meal are the best alternative plant protein sources. From these, we have found that coconut meal based diet basically a very important and having a large amount of protein, different type of amino acids, carbohydrates, and fat contents. It is also a rich source of vitamins and minerals and basically available at very low cost. After extraction of oil from this coconut, it will be available at very low cost. If we talk about the nutritional value, coconut meal is basically a replacer of fish meal and is going to be used in fish industry. It basically contains about 39% carbohydrates, 14 to 23% of protein, about 13% of lipids, and it also contains 4,414 kilocalories per kg gross energy. Here is our experimental animal, that is basically Cyprinus carvio, or you can call it is common carp. It is basically a bottom feeder fish and basically feed on everything of material. If we talk about the methodology, first of all, we will talk about the fish at experimental conditions. Fingerlings of fish were purchased from government fish seed hatchery Manama and were acclimatized to experimental conditions for about 14 days. And after 14 days, fish were fed on their prescribed diet that we have prepared in this experiment. If we talk about the formulation of feed pellets were formulated by the help of different ingredients in which fish meal, fish oil, coconut meal, wheat flour, and different substitutes such as vitamins, minerals, ascorbic acid, etc. were used in this feed. On the other hand, feed pelleting machine was utilized for the preparation of these feed pellets. If we talk about the feeding protocol, feed was fed at the 4% of live wet weight of fishes, and the feeding period was about 2 hours. After the 2 hours, so all the tanks were washed and refilled with tap water. After that, feces from this were collected after about the 3 hours. Here is a basically experimental design in which you can say overall fish tanks, and here is the inner view of fish tank. This is also another inner view having sieve, and this is outer walls. By the help of these walls, we were able to collect the fish feces. Here is the diet composition of all the diets, in which you can see that the coconut meal was as replacer of fish meal, and L-malic acid was used as supplemented oilseed meal-based diet. On the other hand, sunflower, wheat flour, rice polish, fish oil, vitamin premix, mineral premix, ascorbic acid, and chromic oxide was also incorporated in this diet. If we talk about the parameters, we have analyzed carcass composition, growth performance, nutrient digestibility, and hematological indices with mineral absorption. If we talk about the growth study, we have analyzed total fish weight gain, weight gain percentage, FCR and SCR by the help of these standard formula. On the other hand, chemical analysis of carcass, feed, and feces were done by the help of different instruments in which moisture was done by the help of oven drying method, crude fiber was done by the help of loss on ignition. Crude protein was analyzed by the help of microarraydal operators, whereas a crude fat was analyzed by the help of SOX-led system. Ash was detected by ignition at 650 degrees centigrade. Gas energy was analyzed by the help of oxygen bomb calorimeter, and mineral absorption was done by the help of atomic absorption spectrophotometer. On the other hand, phosphorus, sodium, potassium, and chromic oxide were analyzed by the help of US-WIS 2001 spectrophotometer, flame photometer, as I have told you. Whereas at the end, apparent digestibility coefficient was calculated by the help of this formula. If we talk about the chemical composition of blood samples, all the blood parameters in which RBCs, WBCs, hematocrit, HB, MCV, MCV-LC, all the parameters were calculated in this study. If we talk about the statistical analysis, all the minerals, hematological parameters, nutrients, and growth parameters were subjected to one way of ANOVA by the help of Tukey's honesty significance test. If we talk about the results, from results, we found that the diet number 4, having 3 gram per kg of L-malic acid, showing the best results in terms of weight gain, highest weight gain percentage, better FCR, highest SGR, and survival rate. Similarly, in body composition, we have again found that there was highest protein contents, fat contents, and grass energy in fish that were fed on test diet number 4, having 3 gram per kg of L-malic acid. Similarly, when we analyze the nutrient digestibility, we have also found that crude fat was highly digestible at test diet number 4, having 4 gram per L-malic acid. On the other hand, highest protein and grass energy were analyzed in the fish that were fed on test diet number 3, having 3 gram per kg of L-malic acid. If we talk about the hematological indices, again we have found that the fish that were fed on test diet number 4, having 3 gram per kg of L-malic acid, showing the better number of RBCs, WBCs, better platelet, HB, and PCB volume, and the neutrophils. At the end, if we talk about the minerals, we have also found that the fish that were fed on test diet number 4, having 3 gram per kg of L-malic acid, was best diet in terms of major mineral absorption, in which calcium, sodium, potassium, and phosphorus were absorbed in fish on test diet number 4, whereas copper was absorbed on control diet. On the other hand, if we talk about the iron and zinc that were absorbed on test diet number 3, and magnesium was absorbed on test number 5. If we talk about the conclusion, in present study, we can demonstrate that dietary L-malic acid might have a positive impact on performance and strength of common carb through improving absorption of essential nutrients and minerals, conferring better growth when fed on optimum levels of organic acid and oilseed meal-based diet. From this study, we were able to find that the L-malic acid at the rate of 3 gram per kg supplemented in coconut meal-based diet is the better feed for all fishes, having best growth and other parameters. So thank you for your patience and your time.
Thank you, Dr. Mohammad Mudassir Shahzad, for your productive presentation. Our next presenter is Zarsha Hassan from Government College University of Faisalabad, and her title is Carissa papaya and 17-alpha methyl testosterone as reproductive inhibitor in Oreochromis niloticus. Please, Zarsha Hassan.
Everyone, this is Zarsha Hassan, and the topic of my research is Carica papaya and 17-alpha methyl testosterone as reproductive inhibitor in Oreochromis niloticus. The study generally aimed to check the effectiveness of papaya seeds meal and 17-alpha-methyl testosterone on reproductive performance, growth parameters, and approximate body composition of Oreochromis niloticus. For this purpose, a trial of 3 months was conducted. Agriculture has provided mankind with aquatic and terrestrial food sources for about 5,000 years. Pakistan occupies 34th rank among aquaculture producer countries of the world. With a coastal line of about 1120 km, Pakistan has enough fishery resources that remain to be developed. Tilapia is most frequently cultivated species of fish, coming second only to the carps in terms of importance. The problems regarding tilapia: unwanted and early spawning. How to control unwanted spawning. Monosex culture techniques is one of the solution to control the unwanted and early spawning by means of natural resources and synthetic. First of all, fry were collected from the hatchery and acclimatized for 3 to 4 days. After that, 25 fishes were randomly distributed in 5 different aquariums with one replica. This trial was completed in 2 phases. The first one is treatment phase and the second one is rearing phase. In treatment phase, 4 treatment dyes were given with one control group. 2 of them comprises 2 different levels of papaya seeds meal, 6 and 7 gram per kg, and 2 of them comprises 17 alpha methyl testosterone in 2 different levels like 60 mg and 70 mg per kg. In rearing phase, all the fishes were fed on controlled diet, and throughout the experiment all the water quality parameters were monitored and DO was maintained through capillary system. All the growth parameters like increase in weight, increase in length were calculated on fortnight basis. They were mixed together with different inclusion levels of papaya seeds meal and 17 alpha methyl testosterone to form the dough, and after that a minute size pellets were created. Feed was calculated by linear formulation method used. At the end of trial, fish were dissected randomly from each aquarium to determine their sex and to calculate the gonadal history like GSI and HSI of fishes. Feed and body meat was homogenized utilizing a mortar and pestle and examined chemically by AOAC procedures. Like crude fat was determined by Soctic system and crude protein by microjuldan analysis, ash analysis by electric furnace, and dry matter by oven drying. Statistically, it was shown that the higher weight gain was observed by PSM6 and the lowest weight gain was observed by MT70 mg per kg. In case of length increment at 4th fortnight, the highest weight gain was observed by PSM6. In case of length increment, highest increase in length was calculated in PSM 6 gram per kg at 4th fortnight, and SGR indicates that papaya seeds meal enhance the growth rate and PSM 6 gram is better suited for the better growth. And the value of PER were remained almost constant in all treatments. Data suggested that 17 alpha methyl testosterone successfully produced a higher number of males as compared to the papaya seeds meal. And in case of papaya seeds meal, large number of sterile Oreochromis niloticus were produced. Bar graphs of GSI of males and females indicated that low value of GSI and HSI suggested that fish cannot breed. PSM supplementation in experimental diet showed higher growth performance as compared to other experimental groups. Papaya leaves and seeds comprises many phytochemicals like alkaloids, flavonoids, terpenoids, which all promotes higher growth and helps the muscles to work properly. 17 alpha methyl testosterone successfully produced male Oreochromis niloticus as the male percentage was high in empty treated group as compared to the other control and PSM groups. Plant source papaya seeds meal successfully affected the reproductive performance of Oreochromis niloticus. Treatment that fed on PSM 6 gram per kg and 7 gram per kg produced numerous sterile Oreochromis niloticus. Low gonadosomatic index GSI indicates the underdevelopment of gonads, due to which fish cannot breed. Results of current studies showed that in addition to the significant effects that dietary variations in 17-alpha-methyl testosterone and carica papaya had on development and ability of Oreochromis niloticus to produce, carica papaya caused sterility in Oreochromis nilotica and significantly increased growth rate and protein retention in the fish as compared to 17-alpha-methyl testosterone. So I suggest that plant sources are preferable to artificial means of controlling Oreochromis niloticus uncontrolled proliferation since they are affordable, biodegradable and readily available. Thank you.
Thank you, Zarsha Hassan, for your astounding presentation. Iman Naeem from Government College University of Faisalabad is going to present her abstract, and title of her abstract is Efficacy of Different Types of Biochar on Growth, Performance and Body Composition of Stenophrangodon idella fingerlings. Please, Iman Naeem.
Bismillahirrahmanirrahim. Assalamu alaikum to all. I am Iman Naeem. I am the student of MPhil Zoology, Government College University, Faisalabad. First of all, I will thank to Applied Zoological Society for providing me a platform to present my research work. So today I will discuss the title of my research, the efficacy of different types of biochar on growth, performance and body composition of Stenophrangodon idella fingerlings. Introduction. Aquaculture. Almost 50% of world population are suffering from malnutrition, so aquaculture is the fastest growing food producing sector. It is the farming of aquatic animals, for example, fish, crustaceans, plants as well. Aquaculture accounts for 46% of global production and 52% of all fish consumed by humans. It will produce 114.5 million tons of food annually by the year 2025. Fish is the good source of protein. For the better production of fish, fish meal is used. Fish meal is a good source of essential amino acids, digestible nutrients, high palatability and free of anti-nutritional factors. Fish meal prices are rising continuously, so aquaculture is looking for alternative and environment friendly sources. The fish used in my research work is Stenophrangodon idella, which is commonly known as grass carp. It is freshwater herbivorous fish, rich in protein and famous for fast growth. The plant meal I will discuss in my research work is sunflower meal, which is the best alternative to fish meal. The cheapest source and almost 45 to 48% protein are present in it. It has less anti-nutritional factors. Various supplements have been employed as additive in fish feed to promote the growth and development of fish. Biochar is the carbon-rich solid derived from carbon biomass, produced by pyrolysis from various types of biomass in a low oxygen thermal process, the temperature ranging from 350 to 1000 degrees Celsius. It is the precursor of activated charcoal. It promotes the growth and survival rate of fish. So, the aim of my research was to determine the effect of different types of biochar on growth performance and body composition supplemented with sunflower meal based diet on grass carp. The methodology I used for conducting my research work was I took grass carp fingerlings from Fish Seed Hatchery, Satyana Road, Faisalabad. I acclimatized them from 2 weeks. Then I prepared diet supplemented with biochar. After 60 days of trial, I analyzed growth and body composition. I also conducted statistical analysis, that is the analysis of variance, one-way ANOVA. Here are the ingredients composition of test diets. I took 7 test diets and the first test diet is control. Test diet 2 contain cotton stick biochar, test diet 3 wheat straw biochar, test diet 4 corn cob biochar, test diet 5 house waste biochar, test diet 6 grass biochar, test diet 7 green waste biochar. I took the same amount of all these biochars, that is 20 mg per kg. Then I analyzed growth study and body composition study. I used standard formulas for weight gain percentage, FCR and SGR. And for body composition, I used Kazadal apparatus for crude protein analysis, Soxhlet apparatus for crude fat analysis, electric furnace for ash, and oxygen bomb calorimeter for gross energy analysis. Here comes the results of my research study. Growth parameters of stenopharyngodon iodella fed on biochar supplemented with sunflower meal based diet. All the test diets have significant difference with the control group. So it is concluded that supplementation of biochar 20 mg per kg of corn cob biochar, which is test diet 4, have best results for growth parameters. Then body composition analysis of stenopharyngodon iodella fingerlings fed on biochar supplemented with sunflower meal based diet. All the test diets have significant difference with the control group. Test diet 4 gave best results, which is supplemented with corn cob biochar 20 mg per kg. Then conclusion. The results indicated that growth performance and carcass composition of grass carp fingerlings were improved with 2% corn cob biochar, but house waste biochar proved negative effects on both parameters. Thank you for your attention.
Thank you, Iman Naeem, for your descriptive talk. Our last presenter is Laiba Amin from Government College University of Faisalabad, and her title of presentation is Peanut Husk and Leaf Fumitory Extract as Antioxidant and Antimicrobial Agents. Please, Laiba Amin.
Assalamualaikum everyone. First of all, I would like to thank all the present committee of ICAZ-2022 for providing my MPhil research work to the Scientific Society platform. My name is Laiba Amin from the Department of Zoology, Government College University, Faisalabad, and topic of my research work is peanut husk and leaf fumitory extract as antioxidant and antimicrobial agents. First of all, introduction. Natural products have therapeutic potential against various diseases such as inflammations and cancer. Since ancient times, natural products have been utilized to prevent and cure microbial infection, developing the antibiotic drugs. Peanut husk as a natural product is effective for many diseases. Traditional Chinese medicines is utilized to provide the peanut skins to cure some illness. to cure many illnesses. Leaf fumigacy belongs to family Fumigaceae, includes small branching animal herbs, helps to create many diseases. The aims and objective of my study is that to perform phytochemical analysis of Fumigaria parviflora and Ehrlichia hypogaea extract. Here, the Fumigaria parviflora is the scientific name of the fine leaf fumigatory tree, and Ehrlichia hypogaea is the scientific name of peanut husk. To investigate an antioxidant potential of both these plant extract. Third, isolation and characterization of strains to check antimicrobial activity. Fourth, to examine the antibacterial activity against the selected strain. Methodology. In my present study, I have been collected the plant's parts from the Faisal bud. Then I dried it under the shade and grinded it into the powder form, and then have been subjected to the green extraction method. The green extraction then concentrated through rotary evaporator. The prepared extract then used for different analysis. First of all, I have conducted the phytochemical analysis in order to check the presence of alkaloid test, saponins test, phenolic test, flavonoid test, and steroid test. In order to check the antibacterial activity of plants extracts against the selected strain have been performed through well diffusion method. Radical quenching activity of the plants have been investigated through DPPH assay. The results, if I talk about my results, Fumigaria parviflora and Ehrlichia hypogaea extract shows the presence of phytochemicals such as flavonoids, saponins, steroids, tannins, and alkaloids. Both extracts, if we will talk about the antibacterial, then both extracts shows the antibacterial activity as well. But the Fumigaria parviflora shows the best or more potent against the Bacillus cereus as compared to the other selected strains. The mean plot representations of the zone of inhibition of Fumigaria parviflora against Bacillus cereus has been in the diagram or in the graph. And the comparative graph of antibacterial activity of Ehrlichia hypogaea and Fumigaria parviflora against Bacillus cereus also shown in the graph. Here, the antibacterial activity of Fumigaria parviflora against different strains has been shown in the graph, and the Bacillus cereus shows the highest potency or more potent results as compared to the E. coli, Pseudomonas aeruginosa, S. aureus, Salmonella enterica, Aromonas hydrophila, and Pasturula multisuda. If we talk about the Ehrlichia hypogaea, that was the peanut husk. Peanut husk shows the more potent and more antibacterial activity as compared to that of the Fumigaria parviflora, because the more ability to kill the bacteria, and the inhibition zone of Ehrlichia hypogaea is more potent against the E. coli bacterial strain. Mean plot representation of the zone of inhibition of Ehrlichia hypogaea against E. coli has been shown in the graph. Comparative graph of the antibacterial activity of Ehrlichia hypogaea and Fumigaria parviflora against E. coli has been shown in the graph, and it shows the more potency of the Ehrlichia hypogaea extract as compared to that of the Fumigaria parviflora. The antibacterial activity of peanut, that was Ehrlichia hypogaea, husk extract against the different strains has been shown in the graph. The E. coli shows the more potency of antibacterial activity as compared to the other selected strain, that was the Bacillus cereus, Pseudomonas aeruginosa, Salmonella enterica, S. aureus, and Aromonas hydrophila, and Pasturula multisuda. Both extracts shows the radical quenching activity, but the peanut husk shows the higher IC50 value, that is 3.490, as compared to that of the IC50 value of, which is near to the IC50 value of the ascorbic acid, that was 3.37 microgram per ml, which shows the Ehrlichia hypogaea have higher radical quenching activity, which is near to our standard ascorbic acid as compared to that of the Fumigaria parviflora. Here, the graph will show quenching activity of peanut husk and the Fumigaria. The ascorbic acid shows the blue line, the peanut husk shows the red line and the familiar shows the blue line more closer to the red line, which shows that the ascorbic acid, which is our standard, have more potency or have a more radical scrunching activity, and our peanut husk is more also have the more. Based on our results presented above, it was concluded from this study that according to biological assessment, it is concluded that both extract, that is Arachis hypogaea and Fumaria parviflora, are more efficient recording biological activities. A major antimicrobacterial and antioxidant agents, both extract provide us opportunity to learn more about them in the field of herbal medicine. It has been determined that Arachis hypogaea and Fumaria parviflora are the source of medicinally active ingredients and have a variety of pharmacological effects. Therefore, it is exciting to discover new treatments using these substances. Moreover, it has been discovered that they have a strong antibacterial properties and can be utilized to isolate antimicrobial medicines. These have considerable antioxidant potential as well. These results recommended that peanut husk, Arachis hypogaea, have good antioxidant properties and can be used instead of ascorbic acid. These results also recommended that there is a need to investigate the biological properties of other fruit peels that are discarded. Thank you for your attention.
Thank you, Laiba Amin, for your productive talk. All the oral presentation session of ICAZ 2022 has ended, and I'm thankful to all the presenters deliberating in various fields of applied zoological sciences. All their constructive intuitions gave us new perspective of significant advancement in biological sciences. Dear presenters, all of your presentation will be uploaded on official YouTube channel of Applied Zoological Society of Pakistan. After their evaluations, awards will be provided to all the winners during ICAZ 2023. Thanks for your participation and attention. Stay tuned with AZSP. Good night. Stay blessed.